Assalamu alaikum everyone. Today we are going to talk about osteoid, matrix vesicles, and some of the bone cells. Osteoid basically is the unmineralized, newly deposited layer of bone matrix, and this is the matrix that is produced by the bone forming cells, which are the osteoblasts. So osteoid basically is the bone matrix that is secreted by the osteoblasts. Any surface of the bone where active bone formation is occurring would be covered by a layer of newly deposited unmineralized bone matrix, which has not yet mineralized, called the osteoid. So when the bone forming cells, that is the osteoblasts, they start forming the bone, they secrete the bone in the form of an unmineralized matrix, and that matrix basically consists of type 1 collagen fibrils arranged more or less parallel to the bone surface, embedded in a complex ground substance of proteoglycans, glycoproteins, and other protein molecules, and this layer is called the osteoid, as long as it is unmineralized. When the osteoid reaches a certain thickness, that is about 5 to 10 micrometers, it gains the maturity which is conductive to mineralization, and then it starts mineralizing into the mature bone. When the alveolar bone is first formed, it is in the form of an unmineralized matrix, and within that unmineralized matrix, the initial mineralization is controlled by the bone forming cells themselves, which are the osteoblasts. These bone forming cells, alongside secreting the bone matrix, they also secrete membrane bounded vesicles that are called the matrix vesicles, and these vesicles have the first crystal for the initiation of mineralization. These matrix vesicles, when they bud off into the osteoid or the unmineralized bone matrix, they are bounded by the cell membrane initially, and they have the first crystal for the initiation of crystallization or mineralization. As the crystal within the confines of the matrix vesicle grows, it eventually comes out of the confines of the membrane of the matrix vesicle. And then when it comes out of the confines of the matrix vesicle within the unmineralized matrix of the bone, there it forms a seed around which further mineralization could occur. And the process by which the further mineralization occurs around numerous seeds from the numerous matrix vesicles within the bone matrix is called the epitaxy. Epitaxy basically means that a single crystal would grow on top of a crystalline substrate. So basically a substrate of crystal is needed for the crystallization or mineralization to continue or to initiate, and that is provided by the matrix vesicle. So osteoid basically, just like predentine, is a pre-bone or unmineralized matrix that the osteoblasts initially secrete, and as long as it is unmineralized, it is called the osteoid. जब वो mineralize हो जाता है तो तब तक osteoblast एक नया bone matrix की layer secrete कर चुके होते हैं, वो osteoid बन जाता है. जब वो mineralize हो जाता है तब तक नई osteoid की layer secrete हो चुकी होती है, and so on and so forth, बिल्कुल वैसे ही जैसे dentine में होता था. Matrix vesicles, as I've said, by definition, are small membrane bounded structures that bud off from the cell forming the bone, the osteoblast, and they form an independent unit within the first formed organic matrix of the hard tissues. The first morphological evidence of a crystallite is seen within these vesicles. These vesicles basically, as I've said, that they provide a micro environment in which the proposed mechanisms for initial mineralization exist. So basically, these matrix vesicles, they're coming off from the osteoblast, and they are providing an environment which would help in the initiation of mineralization. So definitely they have certain components within their cell membrane or cytoplasm. These matrix vesicles basically consist of alkaline phosphatases, calcium ATPases, metalloproteinases, proteoglycans, and anionic phospholipids. These components bind calcium and inorganic phosphate, thereby forming the first calcium inorganic phospholipid complex, which forms the basis for the initiation of mineralization. And then once this mineral comes out of the confines of the membrane of the vesicle, it acts as a seed within the unmineralized bone matrix, and over that seed more and more mineral is deposited, and eventually numerous adjacent seeds, or you could say numerous adjacent mineralization foci, are created within the bone matrix. And hence when all of these foci, they coalesce together or they fuse together, they give rise to a mineralized, homogeneous mineralized bone. This figure here shows the matrix vesicles. These arrows are pointing towards the cell membrane bounded vesicles called the matrix vesicles. अगर matrix vesicle को हम freeze करके इसका structure देखें तो it has a lot of intramembranous particles that are thought to be to represent the enzymes that are needed for the mineralization. This figure here shows the cell membrane of the vesicle, which has the calcium ATPase activity. So basically this membrane bounded structure has all the elements that are needed for the first mineralization, or the first mineral, or the first crystallite to develop. And once that crystallite, it comes out of the confines of this membrane, it acts as a mineralization seed, or the mineralization foci, over which mineralization then continues. Matrix vesicle, this figure here shows the matrix vesicle surrounded by numerous collagenous fibrils. Some of them are mineralized and some of them are unmineralized. These are the collagen fibrils that you can see, and then you can see numerous matrix vesicles. These matrix vesicles, they form the mineralization foci. So imagine if you have all of these mineralization foci, and all of these crystals, they grow, and eventually this whole region would be mineralized. This is how the mineralization happened. This figure here shows the mineralization foci. अब वहाँ पे आपको confines के अंदर नज़र आ रहा था मैट्रिक्स वेसिकल, अभी रप्चर नहीं हुआ था. यहाँ पे मैट्रिक्स वेसिकल रप्चर हो चुका है और ये क्रिस्टलाइट्स confines of the membrane से बाहर निकल आए हैं. So these things that you can see here are the mineralization foci. So when all of these mineralization foci would grow and eventually fuse, ये form ले लेंगे. They would form a mineralized bone matrix. And by the time this region mineralizes, यहाँ पे नया osteoid secrete हो चुका होता है, और फिर वो mineralize करते हैं, और osteoblasts eventually फिर यहाँ पे osteoid secrete करते हैं, and so on and so forth. ठीक है. This figure here shows the linear profiles among the calcified collagen fibrils. These arrows are pointing towards the numerous mineralization foci present between the numerous collagen fibers, because obviously you know that bone consists of mainly collagen fibers. It has 67% mineral and 28% is the collagen fiber. Then we are going to talk about the bone cells. In today's lecture, we are going to only cover the bone forming cells. However, bone not only consists of the bone forming cells, but it also has the bone resorbing cells, which are the osteoclasts, the bone forming cells, the osteoblasts, and the mature bone cells, or the cells entrapped within the matrix, the osteocytes. The actively synthesizing cells or the actively secreting cells of the bone are the osteoblasts. These cells are mononucleated cells that synthesize the organic matrix of the bone. When these osteoblasts, they are actively secreting the bone, they are cuboidal plum-shaped cells. And when they're inactive and not actively secreting the bone, they look like flattened cells covering the surface of the bone. However, the active plump cuboidal osteoblasts exhibit abundant amount of well-developed protein synthesizing organelles, obviously because these are synthesizing cells, so they have to secrete the bone matrix, so for that they need the protein synthesizing organelles. And the main secretory product of these osteoblasts is type 1 collagen, which makes the dominant component of the organic matrix of the bone, type 5 collagen, proteoglycans, and numerous non-collagenous proteins, the sialoprotein and the osteoprotein, and all of these non-collagenous proteins basically regulate the mineral deposition. And the rest of the non-collagenous proteins that the bone consists of, like osteonectin and tenascin and so on. So these osteoblasts basically are pluripotent cells, and they're coming from an osteoprogenitor cell. So a single osteoprogenitor cell could differentiate into an actively bone synthesizing cell, the osteoblast, and these osteoblasts, they are destined to secrete all of the bone matrix, which eventually mineralizes into the mature bone. And during this process of secreting the bone matrix, these osteoblasts, they could get entrapped within the matrix that they secrete, just like the cementoblasts. And once they get entrapped within their own matrix that they are secreting, they lose the protein synthesizing organelles, and they are no more the actively synthesizing cells. And there they reside within the houses called the lacunae. And these inactive cells are then called the osteocytes. So the osteoprogenitor cell differentiates into an osteoblast, and an osteoblast, when it gets entrapped within its own matrix, it becomes an osteocyte. This is how an osteoblast typically looks like. An active osteoblast, a plump cell with a single nucleus, and this nucleus is not present in the center of the cell. Basically, this nucleus has an eccentric position. What does eccentric mean? The nucleus is off the center. जैसे कि आपको बहुत clearly इस figure में नजर आ रहा है, it is not in the center, it is off the center. This figure here also clearly shows the eccentric location of the nucleus. This is a section of the bone where you can see numerous osteoblasts lined up and secreting the bone matrix. If you zoom a few of these osteoblasts, you would see cuboidal plum-shaped cells with an eccentric basophilic nucleus having the rest of the pale eosinophilic cytoplasm. So this is what the osteoblasts look like when they are actively secreting the bone matrix. This figure here clearly shows the difference between the actively secreting bone cells, the osteoblasts, and the resting bone cells, or the resting osteoblasts. When they're resting, they're called the bone lining cells. So on this side, you have these plump cuboidal cells that are actively secreting the bone. So this region has the active synthesis of the bone. So here the cells lining are the cuboidal plump cells, the osteoblasts, and on this side no bone formation is occurring actively. So these cells are flattened cells and they cover the bone surface, and they are called the bone lining cells. So osteoblasts, as we've said, that when they're actively secreting the bone matrix, they look like cuboidal plump cells with an eccentric nucleus, and obviously they have lots of protein synthesizing organelles, and they are destined to secrete all of the bone matrix. So what would they produce? They would produce everything that the bone has, the non-collagenous matrix proteins, and all of the collagen fibers, the type 1, type 5 and type 3 collagen fibers. Just means the type 1 is predominant. So these osteoblasts, when they secrete these components of the bone matrix, they secrete the type 1 collagen just like the fibroblast was secreting the collagen. The process is exactly the same. Once the osteoblast has produced the collagen, these collagen fibers, they are packed into secretory granules, which are typically elongated and electron dense. And along with these collagen fibrils, it is believed that some non-collagenous matrix proteins are also packed into these secretory granules, and they are released against the surface of the cell opposed to the forming bone. Once these molecules have been secreted into the forming bone, these molecules assemble there as fibrils, and they accumulate as a layer of unmineralized or uncalcified matrix, because there's no mineral yet, and it is called as the prebone or the osteoid. So if I draw the whole thing that I've just talked about here, it would look like this: cuboidal cells with an eccentric nucleus, cuboidal cell with an eccentric nucleus. It secretes the produces the collagen fibrils, and it is packed into elongated secretory granules. So these secretory granules, they would exit the osteoblast the secretory granule will obviously exit the osteoblast, and it would accumulate or exocytose all of the contents that are present within it. What does the secretory granule have? As we've just said, the secretory granule has the collagen, and it also has the non-collagenous matrix proteins. So the granules, when they empty themselves outside the cell, they accumulate here in the forming bone region as a layer of uncalcified matrix, which is called the osteoid. And we know that the uncalcified matrix comprises of the collagen fibrils embedded within the ground substance. So this is how we get the osteoid. Once we have got the osteoid and it attains about 5 to 10 micrometer thickness, the matrix vesicles, they come into play, and they start mineralizing the osteoid into the mature bone. This figure here shows the pre-osteoblast and the osteoblast. The pre-osteoblast is a comparatively elongated cell, and the osteoblast looks like a plump cuboidal cell with an eccentric nucleus. Along with secreting the components of the bone matrix, the osteoblasts also secrete a number of cytokines and growth factors that basically help regulate the cellular function of the bone and the formation of the bone. And these cytokines and growth factors include bone morphogenetic protein 2, 7, transforming growth factor beta, insulin-like growth factor 1, 2, platelet-derived growth factors, and fibroblastic growth factors. And what do these growth factors and cytokines do? They regulate the cellular function of the bone, and they regulate the formation of the bone. But these cytokines and growth hormones, their secretion is under hormonal control. So there are certain hormones that control the secretion of these cytokines and growth hormones from the osteoblasts. And those hormones include the leptins, the calcitonin, the estrogen, the glucocorticoids, the parathyroid hormone, and the 1,25-dihydroxyvitamin D. So these hormones would control the secretion of these cytokines. So what does that mean? That when these hormones would control that when the cytokines are needed from the osteoblast to promote bone formation, and when these are not needed, or when the bone formation needs to be inhibited. So basically the bone formation or the regulation of cellular function and bone formation is under hormonal control. These hormones affect bone most likely. These hormones that affect the bone formation most likely work primarily through altering the cytokines and growth factors. जैसे कि मैंने अभी बोला कि जो hormones हैं वो basically how do they work? They basically control the secretion of cytokines and growth factors, and that is how they are affecting the bone. Out of these hormones, the parathyroid hormone and the vitamin D have a dual function. What does that mean? That these two hormones have the ability to enhance the bone resorption when they're present in high concentration, when they're released in high concentration. But at the same time, at lower concentrations, these two hormones support the bone formation. So according to their concentration, the bone formation and the bone resorption could be enhanced or supported. Then the calcitonin and estrogen basically inhibit the resorption of the bone, whereas the glucocorticoids, they inhibit the resorption and formation, but primarily they inhibit the formation. Leptin is basically a circulating hormone that is produced by the adipocytes, and this hormone has the ability to promote and inhibit the differentiation of osteoclasts. So by promoting and inhibiting the differentiation of these bone resorbing cells, this hormone has the ability to either promote resorption or inhibit resorption depending upon the stimulus. It may work locally to promote the differentiation of the osteoprogenitor cells and hence stimulating the osteoblasts to make new bone. So this hormone has a function in inhibiting and promoting the osteoclast differentiation, and also it has the ability to promote or stimulate the osteoblast differentiation to help in the formation of new bone. So this is how these different hormones are actually controlling the bone formation and bone resorption by controlling the secretion of the cytokines and growth factors. So this is how your bone formation and cellular function of the bone is being regulated by the hormones actually. So once the osteoblasts have secreted all of the bone matrix, they form a layer over the bone surface, and as they have secreted all of the bone, they no longer remain the active bone forming cells. They eventually lose their protein synthesizing organelles, and there on the bone surface they attain a flattened shape rather than cuboidal plump shape, and their main function over the bone surface is to act as a barrier to control the influx and outflux of certain ions or minerals, or you could say molecules, into and out of the bone. These osteoblasts do not have any junction complexes with adjacent cells. However, they are connected to each other through gap junctions, and these cells are called the bone lining cells, because now they're not actively forming the bone, they're no more the forming cells, and they have substantially achieved a flattened appearance, as I said, by losing their protein synthesizing organelles. They extend through the entire surface of the bone, and they are called the bone lining cells. They're connected to each other through gap junctions, and they're also connected to the cells within the matrix of the bone, the osteocyte. So they basically create a very extensive network that helps to control the mineral hemostasis of the bone, and it ensures the vitality of the bone by acting as a site for mineral exchange between the bone and the blood, because the lining cells are communicating with each other, and they are also communicating with the cells within the bone matrix, which is the osteocyte, through gap junction. So they are at a very ideal place to function as the cells that maintain the mineral hemostasis, and because they are lining the cell surface of the bone and they are controlling the influx and the outflux of certain ions and molecules, so they act as a primary site for the exchange of mineral between the bone and the blood. So this is how the osteoblasts function and how they look like, what do they produce, and what the active osteoblast looks like, how the inactive osteoblast functions and looks like, and where it is present, how these osteoblasts produce the bone matrix, which is initially in the form of an unmineralized calcified layer consisting only of collagen embedded in the ground substance, and then that osteoid eventually mineralizes with the help of the matrix vesicles, which are also produced by the bone forming cells, the osteoblasts, and then eventually the osteoid layer or the unmineralized uncalcified bone mineralizes, and as the certain amount of bone mineralizes, the next osteoid layer has been secreted by then, फिर वो mineralize होती है, and by the time that has mineralized, the next osteoid layer is already there, and the process goes on until all of the bone has been formed. So this was all about the bone forming cells, the osteoid, and the matrix vesicles. The definition of osteoid and the composition of osteoid is very important. The matrix vesicles, basically their definition and the components of the matrix vesicle is very important. And then osteoblasts के अंदर तो obviously bone forming cells हैं. You could be asked any question about the osteoblasts. The next cells that we're going to talk about is the bone, mature bone cells. Basically the cells that get entrapped within the bone matrix during the secretion of the bone, the osteocytes. Osteocytes और osteoclasts के बारे में हम next lecture में बात करेंगे इंशाल्लाह. This is it for today. I am going to see you guys in the question and answer session at 11 o'clock inshallah. Thank you and Allah Hafiz.